Hello everyone, you are watching Blackboard and welcome to our microbiology playlist. In the previous video, we have discussed what a bacteria actually is. You can find the video link in the description below or in the related videos icon. You know that an embryo size is smaller than a grain of rice? In only 9 months, an embryo is grown up to the size of a watermelon. But do you know how much time bacteria take for their growth? Well, when we talk about bacterial growth, it doesn't mean to increase the size. It means to increase the number of bacteria. In today's video, we are going to learn how bacteria grow their population. What are the steps? What are their necessities for growth? Also, how are they divided into different classes according to their different needs? So stay tuned everyone. Bacteria reproduce by binary fission, in which cells replicate their DNA, the cytoplasmic membrane elongates, and the two DNA molecules separate. The membrane invaginates and cross-wall is formed completely. In this way, one parent cell divides to form two daughter cells, and the process continues till the required amount of population is achieved. But wait, a question came to my mind. Why bacteria chose to be replicated by binary fission only? Because binary fission needs only one parent. There is no need to waste time in mating. So a single bacterium produces many new bacteria in a limited amount of time. Also, daughter cells possess the same characteristics as their parents. Now, we all are thinking, isn't it so harmful for humans that bacteria divide at such a fast pace? Well, the nature has all the solutions. The fatal drawback of binary fission is that if a species cannot survive some changes in the environment, the whole population is wiped out. The doubling time of Escherichia coli is 20 minutes. And for Mycobacterium tuberculosis, it's about 18 hours. So, this generation time varies with bacteria. Let's come to the growth cycle of bacteria that has 4 major phases. We'll see it through a graphical representation, where on vertical axis we have number of bacteria and on horizontal axis we have time. Number 1 is the lag phase, during which vigorous metabolic activity occurs, but cells do not divide. In this phase, bacteria are actually preparing themselves for division. Number 2 is logarithmic or exponential phase, in which rapid cell division occurs. Many antibiotics are effective during this phase. If you have any confusion about which phase comes first, the lag or the log phase, remember, A always comes before O in the alphabets. So, the cycle starts with the lag phase. Coming to the third phase, which is called the stationary phase, that is having a steady state. Here, the number of new bacteria produced equals the number of bacteria that die, because nutrient depletion and other toxic products slows down the growth of bacteria. But if cells are grown in a special apparatus called chemostat, where fresh nutrients are added and waste products are removed continuously, they can remain in the log phase forever and do not enter the stationary phase. The fourth and the final one is the death phase, in which there is a decline in the number of bacteria. Bacteria require an optimum concentration of oxygen, carbon dioxide, pH, nutrients at an optimum temperature for best growth. In this video, we'll talk about oxygen preferences of the bacteria. Some bacteria use oxygen to grow and some do not. The reason why oxygen is not used by some bacteria is because it's toxic to them. And now we are going to discuss why is that so. In most organisms, including humans and bacteria, when our cells go through cellular respiration, a process cells use for energy generation, a byproduct is formed called superoxide free radical and is toxic because it has an extra electron. Now, many bacteria have superoxide dismutase enzyme that converts the toxic free radical to a less toxic form, hydrogen peroxide. Also, many bacteria have additional enzymes called catalase and peroxidase that converts hydrogen peroxide to water and oxygen. That is not toxic at all. So, on the basis of oxygen requirement, bacteria are divided into 5 different classes. Obligate aerobes. They require oxygen to grow because their energy generation system is dependent on oxygen as the hydrogen acceptor. And they have superoxide dismutase and catalase to convert the toxic products into the non-toxic ones. An example of such bacteria is MYCOBACTERIUM tuberculosis, a causative agent of tuberculosis. Obligate anaerobes. They cannot grow in the presence of oxygen because they lack either superoxide dismutase or catalase, or both. For example, Clostridium tetani. Facultative anaerobes. They grow faster in the presence of oxygen, but they can also grow in the absence of sufficient oxygen. For example, E.COLI. AEROTOLERANT anaerobes. They grow to some extent in air but multiply much more rapidly in a lower oxygen concentration because they have superoxide dismutase but lack catalase. This class of bacteria includes Clostridium histolyticum. Last ones are MICROAEROPHILES. They require oxygen to grow but at a less concentration than atmospheric oxygen. Campylobacter jejuni is one of them. So this was all about today's topic. If you find it interesting, hit the like button, and if you want to know about other optimum conditions for bacterial growth, subscribe my channel and hit the bell icon to stay notified. Thank you.